Good morning, Mom. Good morning, classmates. So today, I am going to discuss about the cell biology of pancreatic cancer. So what is pancreatic cancer, by the way? So pancreatic cancer is a cancer that is found in the pancreas. The pancreas is an organ in the upper part of the abdomen. It helps one digest in the food ingested and also makes hormones such as insulin. Pancreatic cancer can be silent. So smoking is the most important risk factor. So pancreatic cancer arises when cells in the pancreas undergo genetic mutations, leading to uncontrolled growth and the formation of tumors. It's one of the deadliest cancer due to late detection and limited treatment options. So here are some risk factors in pancreatic cancer, which are the cigarette smoking, exposure to industrial chemical and toxic metals, underweight, and obesity, and of course we have the last, which is the fiber diet. And now let us proceed to the signs and symptoms of pancreatic cancer. So first we have the pale and greasy stools, abdominal pain, jaundice, new onset diabetes, change in bowel habit, indigestion, nausea and vomiting, weight loss, abdominal distension, non-cardiac chest pain, shoulder pain, dysphagia, loss of appetite, feeling of weakness, overall discomfort. So now we have the structure and functions of the pancreas. So there are two main functions of pancreas, which are the exocrine, producing digestive enzymes, and the endocrine, regulating blood sugar with hormones like insulin. So understanding its structure and functions in healthy conditions help identify abnormalities in cancerous cell. Next we have the understanding the cellular process in pancreatic cancer development. Normal pancreatic cells undergo controlled growth, division, and death or apoptosis. Maintaining this balance is essential for tissue homeostasis. Healthy pancreatic cells go through specific cellular processes to ensure proper functions. They engage in controlled growth, replicating as needed, and have mechanism for cell death known as apoptosis. So this balance is crucial to uphold tissue stability and ensure the pancreas functions optimally. So when these processes are disrupted, it can lead to various pancreatic disorders or diseases. So in cellular changes in pancreatic cancer development, first we have the genetic mutations and alterations. Mutations in critical genes like KRAS, TP53, and SMED4 can significantly impact cellular processes. KRAS mutations, for example, often result in uncontrolled cell growth signaling, so promoting excessive cell division. TP53, known as the guardian of the genome, helps regulate cell division and can trigger apoptosis if DNA damage is irreparable. So mutation here can disrupt these controls, allowing damaged cells to survive and potentially form tumors. SMAD4 mutations affect the TGF-beta signaling pathway, which plays a role in regulating cell growth and differentiation. When SMAD4 is mutated, this pathway regulation falters, so contributing to uncontrolled cell division and the potential formation of tumors. These mutations collectively contribute to the dysregulation of cellular process, paving the way of the development of various cancers, including pancreatic cancer. Next is a tumor microenvironment. The tumor microenvironment refers to the surroundings in which a tumor exists, consisting of various elements like neighboring cells, or which is the immune cells, and fibroblasts, blood vessels, and the extracellular matrix, or ECM. In pancreatic cancer, this microenvironment is particularly influential. So pancreatic cancer cells interact dynamically with these components. They might stimulate nearby cells to create an environment that supports tumor growth or suppresses system's ability to attack the cancer cells. Additionally, these interactions can promote the formation of new blood vessels, or the angiogenesis, to supply the tumor with nutrients and oxygen, aiding its growth. So, the extracellular matrix, a network of molecules supporting tissue structure, can also be modified by cancer cells to create a favorable environment for their proliferation and invasion into surrounding tissues. Understanding and targeting this complex interplay between cancer cells and their microenvironment is crucial in developing effective treatments for pancreatic cancer, as these interactions significantly impact the tumor's growth, spread, and response to therapy. Last, we have the roles of oncogenes and tumor suppressor genes. Oncogenes and tumor suppressor genes are crucial players in regulating cell growth and preventing cancer. Oncogenes are genes that, when mutated or activated, can promote cancer development. They often encode proteins involved in cell growth, divisions, or survival. When these genes undergo mutations or overexpress, they can drive cells to proliferate excessively or resist programmed cell death or apoptosis. This uncontrolled cell growth can contribute to the formation and progression of cancer. So conversely, tumor suppressors genes act as guardians of the cell, working to inhibit cell growth or trigger cell death when necessary. Mutations in genes can lead to loss or reduction of their function. As a result, the regulatory mechanisms and restrain abnormal cell growth become compromised. Cells may then divide uncontrollably or evade apoptosis, contributing to the development and progression of cancer. Now let's proceed to the molecular mechanism and driving pancreatic cancer cell or cancer growth. First, we have the aberrant signaling pathways. Signaling pathways are intricate communication systems within cells that regulate various functions like growth, survival, and differentiation. When these pathways are disrupted, as seen in the RAS pathway in cancer, it can significantly impact cellular behavior and contribute to cancer progression. The RAS pathway is crucial in regulating cell growth, division, and differentiation mutations, especially in genes like KRAS can lead to the constant activation of this pathway, causing cells to receive continuous signals to grow and divide. This persistent activation of RAS signaling can drive uncontrolled cell proliferation, a hallmark of cancer. Number 2, we have the cell cycle dysregulation. Dysfunctional cell cycle control mechanism caused by gene mutations lead to unregulated cell division, a hallmark of cancer. When key checkpoints fail to ensure proper DNA integrity or replication, damaged cells can divide, leading to the growth of abnormal cells, a fundamental characteristic of tumors. Therapies targeting this faulty checkpoint aim to restore control over cell division in cancer treatment. Number 3, we have the programming in cancer cells. Pancreatic cancer cells have unique metabolic needs due to their rapid growth and proliferation. they often adapt their metabolic pathways to meet this increased energy demand. So, one prominent alteration is a preference for glycolysis, a process where glucose is metabolized to produce energy. Even in the presence of oxygen, this phenomenon known as the Warburg effect allows cancer cells to generate energy rapidly, supporting their rapid growth. Now let's explore the cell biology-based therapeutic approaches. First, we have the targeted therapies in pancreatic cancer. Targeted therapies focus on specific molecules or pathways crucial for cancer growth. In pancreatic cancer, molecules like EGFR or epidermal growth factor receptor or BRAF or BRAF enzyme are potential targets. So drugs inhibiting these molecules aim to disrupt the signaling pathways that drive cancer cell proliferation. However, their efficacy might vary depending on the specific genetic alterations within the tumor cells. Next, we have the immunotherapy and its role in pancreatic cancer. Treatment immunotherapy aims to metabolize or mobilize the body's immune system to recognize and attack cancer cells. In pancreatic cancer, where standard treatment have limitations, immunotherapy has shown promise. Strategies like immune checkpoint inhibitors or specialized cancer vaccines help activate the immune response against pancreatic cancer cells. However, improving early detection method is crucial as pancreatic cancer is often diagnosed at advanced stages. Understanding the intricate dynamic of the tumor microenvironment is another key area of research. So strategies targeting these challenges might involve combination therapies. So, identifying biomarkers for early detection and refining treatments that consider the complex interactions within the tumor microenvironment. So this avenue of research treatment signifies ongoing efforts to develop more effective personalized and comprehensive therapies for pancreatic cancer. And that would be all. Thank you.